evening, everyone, or rather good afternoon. It is not a Saturday. You will notice that usually those who are following my channel, you'll find that my lessons are usually on Saturday. But today I am free, so I'm going to have a lesson with you, and it's a Monday, right? It's a public school holiday. It's a public holiday. So I'm very sure that many of you are having exams at the moment, right? Or rather in where my school is or where my state is, we're having our mid-year exams. So I'm very sure that many of you are unable to join my class live, but it's all right. You can always click the link anytime to view my videos at any time at your convenience. Okay, right. Today, you will notice that today is just chapter 1. We have already gone probably chapter 5 or 6. So I think that most of you will be using it as a revision. All right. Okay. So for those who are viewing, thank you very much. So I shall start straight with the lesson today. If you would like to let me know who you are, just type a comment on the comment chat box here, and I'll be able to see who you are. All right. Okay, let's get right straight to the lesson. Okay. Now, today is 1.2, which is a continuation of 1.1, which is the organization of tissues, plant tissues. So meristematic tissue, I promised you earlier during 1.1 that I'll be making a video. I'll be making a class on this. So today is the class, right? We're doing 1.2, which is meristematic tissues and growth, right? So those who are having an exam soon, which is bio papers next week, so you can use it as a revision. Okay, right. Now, let's start, yeah? So do you know what you're looking at? Okay, what is it that you're looking at? This, I'm very sure that you'll be very familiar with this. You have seen this in Form 4 last year when you did chapter 6, all right? Some of you will be able to realize that you're actually looking at the purple color parts are the nucleus of cells, okay? And this is actually plant cell. So we're actually looking at a slide, okay, that is taken under a picture taken under the microscope of plant cells which are undergoing various stages of mitosis. Okay, right. So you must be wondering, why are we talking about mitosis, whereas our class today is on meristematic tissues. Actually, meristematic tissues are those tissues are actually undergoing what you call different stages of mitosis. So they're actually carrying out cell division. Okay, all right. So this is what you need to learn about meristematic tissues. Their function, the function of meristematic tissues is actually to divide. And when they divide, they produce more cells. And when more cells are produced, what happens is the plant increases in size. Okay, it increases in length in terms of the shoots, and also the root, it becomes longer. Okay, now first of all, let's look at the meristematic tissues. We have already we've gone through in the previous lesson, in the previous video. We have gone through the ground tissues and the different types of what we call permanent tissues. Okay, so this is meristematic tissues, which are only their function is to carry out cell division, and that is to increase the number of cells. Okay, under meristematic tissues, there are actually two groups. Okay, there are two places, two different types of meristematic tissues. So the first type is called apical meristem. That means, what is the meaning of apical? The apical, the word comes from the word apex. Apex means at the tip, at the very end. So in a plant, you will see there are two places which are very end, at the very end, the tip. So one is what we call the shoot. At the tip of the shoot is what we have the shoot apical meristem. And you have you take a look at under the microscope, you will see that this is what it looks like. Okay, this is the very tip of the end of the shoot, where you will see apical meristem and active mitosis is occurring. All right. Okay, at the other end of the plant, you will be able to see the roots. Okay, so here is the root apical meristem, and this is the slide showing the root apical meristem. So the apical meristems are not scattered everywhere. So there is a certain place, and I've already highlighted with a circle. So for the shoot apical meristem, it's at the very tip of the shoot, whereas for the root apical meristem, it's not at the very tip. You notice that it's behind this area. Okay, so it's behind this, what we call the root cap. So you will see the root cap later, yeah, right? And it has a different function. So meristem, root meristem is here. So if you were asked to label, please label this area. Okay. The other group of meristem is what we call the lateral meristem. So just now apical means at the very tip, at the very end. But lateral means actually side. The meaning of lateral means is the side. Okay, so where is the side? Okay, so if you were to slice through the stem, all right, and also the root, you will find that inside the stem, all right, you will have another group of tissues, what we also call cambium, right, what we call vascular cambium and also cork cambium. Okay, so basically these are the names of tissues. So apical meristem, you have the shoot apical meristem and the root apical meristem. But for lateral meristem, there is the cork cambium and the vascular cambium. All right, okay, so all these tissues that we're looking at now, they carry out mitosis, okay, which is a process of cell division to increase the number of cells. So when the number of cells increases, the plant either goes upwards, right? Grows upwards means longer, or the root goes downwards to become longer. And for the cork cambium and vascular cambium, so when they divide, what happens is the stem becomes fatter, all right? Or we call the diameter increases. So that goes also for the roots, not only the shoot, right? The root and the stem and also the roots. Okay, let's go to the next slide, yeah? Okay, some more about the apical meristem. So here, this is the shoot tip, we call the shoot apical meristem, and here is the root apical meristem at the root tip, right? Now, all these cells that you see in the first slide earlier, you see all these being colored, stained purple and all that, they actually show the nucleus. And if you were to magnify, right, you go further, you will be able to see that in the nucleus is actually undergoing different stages of mitosis. So some are probably going under prophase, some at metaphase, some anaphase, and even some are at telophase. Okay, what are the things we need to know about meristematic tissues? Okay, so they consist of undifferentiated cells. All right, that means the cells are not being differentiated to become like xylem or phloem. They don't have any specific jobs to do, but what they actually do is just to divide. They just divide and increase the number of cells for the plant. Okay, and they divide actively by mitosis. Okay, never meiosis, mitosis. Meiosis is only for sexual reproduction, which involve production of gametes. Okay, next, right? So when the meristematic tissues divides, they will cause the shoot to grow vertically upwards because the number of cells increases. So therefore, the shoot will also elongate. The shoot will become longer, and that causes the plant to grow upwards. And that's important, right? You know, the plant has to grow, and it has to receive sunlight so that it can carry out photosynthesis. So growing requires these meristematic tissues to do their job, which is to divide. And at the same time, you find that roots will also carry out mitosis, the root cells, which is the meristematic tissues, the cells, and they grow vertically longer. That makes the root go downwards. And then you will find that the roots will be able to penetrate deeper into the soil to obtain or to absorb more water, okay, to support the plant. Okay, so next slide. Yeah, I don't see any questions. Probably there's not many people joining my class because I'm very sure you are revising for tomorrow's paper. Okay, that's fine. Okay, now lateral meristem. Let's talk about lateral meristem. Okay, so lateral meristem is found at the stem and the root. Okay, so now if you were to cut through, okay, of course this is a very small plant, but if you were to cut through a thicker plant, right, you'll be able to see something like this. So there are two groups of tissue here, which are under lateral meristem. So we call that cork cambium, which is nearer to the epidermis, and another layer which is nearer to the inside, nearer to the pith, is called the vascular cambium. So these two are under lateral meristem. Okay, now what is the function of this lateral meristem, the cord cambium and the vascular cambium. So what they do is when they divide, they also divide by mitosis, yeah? All right, so the number of cells increases. So what happens is it pushes outwards. Okay, the phloem will be, the old phloem will be pushed outwards and the old xylem will be pushed inwards. So the new cells that are formed are called new xylem, we call it secondary xylem, and the new phloem is going to be called secondary phloem. So that means by having this lateral meristem, it increases the number of xylem tissues and phloem tissues. Okay, that's important. When a cell or when a plant grows, it has more demand for water. All right, it has to transport water and food more efficiently. Therefore, they need to have more xylem tissues and also more phloem tissues. So these two cambium tissues, they make new xylem and new phloem, and they are known as secondary xylem and secondary phloem. Okay, later we will see where they are, and you must know how to label them. Okay, next. Now, so here we come to an important part about the meristematic tissues. You need to know that there are actually three zones. All right, at the shoot tip, there are actually three specific zones whereby the cells carry out different function. All right. Now you will see at the very tip of the shoot, you have this shoot apical meristem. And as we learned earlier, the job of this shoot apical meristem is to divide. That means it carries out cell division. Therefore, this zone is called zone of cell division. It's at the very tip, okay, at the very end of the shoot. Now further downwards, okay, after this new cells that are formed by cell division, a while later, right, maybe some weeks later, right, what happens is these cells will become longer. So they'll be pushed downwards into this zone of cell elongation. So at this area, we call the zone of cell elongation, you will find that the cells are actually longer in length compared to the cell division, the zone of cell division. The cells over here in the zone of cell division is actually more squarish, more like we call cuboidal, cube shape, compared to the zone of cell elongation, which has the cells like more of a rectangular shape, longer. All right. Okay, and further down, after these cells have elongated, they will undergo differentiation. Differentiation means not every cell will be the same anymore. Some tissue here will be converted or it will change to become xylem. It will change to become phloem. So therefore you will see in this zone of cell differentiation, there will be xylem, phloem, and some other, you can see the cortex, okay, all this will be formed. So it started as normal, the meristematic tissues that will produce the new cells. Later on, it will become differentiated to perform different jobs. Okay, so you have xylem, phloem, and cortex. So when you see that labeled here, that means this is definitely the zone of cell differentiation. Okay, right. Now, what about the root tip? Root tip, you also have the same exactly three types of what we call the zones. Okay, but they're in reverse order because you always start from the tip. Okay, therefore the zone of cell division is always at the tip. But look carefully, it is not right at the tip. Yeah, this is at the very tip. You have this root cap, which is actually not meristematic tissues. Okay, they don't carry out mitosis, but this root cap has important function, which is actually to protect the root apical meristem that is in the zone of cell division. Okay, because you notice that these roots, they go down and they go into the soil. So into the soil, there are actually pebbles, there are rocks, right? There are very jagged edges of the rocks and sand and all that. So that could injure this delicate root apical meristem. So in order to protect this root apical meristem, you have a layer of, a few layers of these root cap cells, which are going to protect, and also this root cap cells produces a kind of mucus, some kind of a liquid, which is called mucilage, mucilage, to lubricate, to make it like, smoother, so that it's easier for the root tip to penetrate into the soil. Okay, so now after this zone of cell division, you will have all these new cells form. Then later, these cells will undergo cell elongation. They will become longer, just like what I've mentioned for the shoot tip. And then after a while, these cells, which are, rectangular in shape, they will slowly differentiate into different tissues or different cells that perform different functions. So you will see some of them will become the cortex, some will form the root hair cells, okay, and some will become the phloem, some become the xylem. And further up, which is the, further after the cell differentiation, you will have all these permanent tissues which are matured. They're already formed. You have the xylem and the phloem and all these tissues there. Okay, so they are mature tissues, which is after the zone of cell differentiation. All right, okay, so this picture you can find in the textbook. Okay, it's a very important picture that you need to know how to label the different and the three types of zones. Right, don't get it reversed, yeah? We always start from the tip, and we start with cell division, followed by cell elongation, and then cell differentiation. Okay, for the root, it's the other way around. All right, we always start from the tip. Okay, so this is the root cap. So do not label wrongly. There are many students who always label the tip here as the meristem tissue, which is wrong. It is always the root cap at the tip of the root. All right, okay, next. So let's talk about the root tip. Yeah, let me show you a picture that shows the detail. Right now, you can see this of the cells, right? So here is this zone of cell division. You will see that mostly the cells here are cube shape. They're not very long. They're small cells, but with very big nucleus. Okay, so this is the root apical meristem. This is where you label the zone of cell division. All right, now this is what the cells look like. Okay, drawing lah. All right, you can see that all these cells undergo different stages of mitosis. Some have already, you will have the metaphase, some are already telophase, some are still undergoing anaphase, and so on. Okay. Now, this is the root apical meristem. If you see, this is how it looks like. So the cells here are actively dividing through mitosis, right? And they have not undergone differentiation. So these cells are called undifferentiated. Okay, all the cells are exactly the same. They all carry the same function, which is just to divide. And the root cap here, which is not part of the meristem, which is actually at the tip of the meristem, it protects the apical meristem. Okay, so be careful about the labeling of the root cap. And also produces mucilage. Okay, so this is the root cap cells. Okay, further up, you will have this zone of cell elongation. You can see very clearly that these cells are actually rectangular in shape. They're longer. They're longer than and bigger, right? Compared to the cells in the zone of cell division. And further up, you will see that these cells have taken on different roles, different jobs. So we will call this area here is the zone of cell differentiation. They have become xylem, phloem, you can see root hair, right? The cells, some of them, the epidermis here have been modified to have projections, and that is the root hair cell, okay? Right, now next part, I will show you about this. Okay, this is another picture showing the root tip, but this is actual slide taken under the microscope. Just now it's just drawing. All right, so root apical meristem is here. All right, and the same picture here shows you. So if you put the label, it's over here. All right, the cells are undergoing various stages of mitosis. All right, now we look at zone of cell, not cell division, yeah. So this one, zone of cell division is still here. We're talking about the same thing, yeah. Behind the root cap, produces new cells through mitosis. All right, so it's a bit of like continuation. This is actually a picture shown under the microscope of the meristematic tissues undergoing different stages of mitosis. Now, let's remember this is very important. the characteristic of the cells which are in the zone of cell division. So remember this, they have thin cell wall. And sometimes if the question asks you, why do you say that these are meristematic tissues? So here is your answer. You say that these are the characteristics. You see that they have this, probably they are meristematic tissues. Number one, thin cell wall. Number two, large nucleus. You can see the volume taken up by the nucleus. It's very big compared to the entire volume of the cell. Okay, it's more than half of the volume of the cell is taken up by the nucleus. Next, you have dense cytoplasm. Okay, so the cytoplasm is quite dense. So when you draw your cytoplasm, never draw it empty. Yeah? You have to put dots. Dot your cytoplasm to show, right? This is not vacuum, right? And next, another important feature, there is no vacuum, or rather there is small vacuum. Okay, so follow the textbook, your Form 5 textbook. So your textbook says small vacuum, you can follow that. If the textbook says no vacuum, it's also correct. Okay, for the zone of the cells under the zone of cell division. Okay, now how do they elongate? Okay, how do these cells after they are being formed, how do they elongate? All right, so they carry out a process which is called vacuolation. Okay, so what happens is, now cells from the zone of cell division, after they have been formed, they will enlarge. How do they enlarge? They take in water. They absorb water through the process you have learned earlier. This is osmosis. So movement of water, okay, from a region of higher water potential into less or lower water potential is called osmosis. So water goes in. So when water goes in, you will see that the small vacuoles, right? Of course, at first they are very small. Then the vacuole will start to become bigger. So they merge. They merge means they join together. So you see this picture here, the second picture, the picture here, right? You will see here that when water is absorbed into the cell, so the vacuoles become bigger and they join together. This process is called vacuolation. Okay, so when the vacuoles join up, they fuse together to become a bigger vacuole. They expand, right? And we call this a central vacuole. So at the zone of cell elongation, you will be able to see that the vacuoles have joined and the vacuoles are large. So this is an indication that this cell you're looking at is at the zone of cell elongation. Okay, it's bigger. All right. So they form a large central vacuole. So, and because of this, the cell swells, means it becomes bigger, and it elongates because of the pressure. We call the turgor pressure. The water goes in, it gives a pressure. That water pressure is called turgor pressure. Increases on the cell wall, and that causes the cell to become longer. Okay, so now the cell is longer, and this is the zone of cell invasion. Okay, now one more, the last thing. The zone of cell differentiation, how do you recognize is the zone of cell invasion? You will see that inside this zone of cell differentiation, there will be like xylem form, you will see phloem, all right, that is formed in this area. So the cells undergo differentiation because now the cells have specific jobs to do. They are no longer undifferentiated. Each cell will be different from each other. Some of them will become xylem, some become phloem, and so on. Okay, so that they can do different jobs. So one example that I can show you here is you can see this root hair cell, right? So it's also differentiated because it's now have projection. It now has projections here, so that will go deeper into the soil, and that increases the surface area so that it can absorb more water. Okay, so now, so done about these three important zones. So always remember, you need to label the key zones, and you need to give explanation or describe what the cells look like and why do we call it as a zone of cell invasion and so on. Okay, the characteristics of the cells. Okay, now so here is the shoot tip, right? So this is pictures taken under the microscope. So if you were to label a zone of cell division, here it is. Okay, and the next one here is zone of cell elongation. And after that, you will have the zone of cell differentiation. Okay, what about the, this is a diagram form, same thing, right? This is apical meristem, and you have the young leaf here at the side, right? And then whenever you see this is showing xylem and phloem. So this is already in the zone of cell differentiation. Okay, when you see the xylem and phloem, it's already in the zone of cell differentiation. All right, so this is where you label your 3 zones. Okay, right. Now, another important skill that you have is you must know how to recognize the drawing that's given to you or the drawing that's in the question paper. Are you looking at a monocot stem or a monocot root, or are you looking at a udicot stem or udicot root? You need to know. Okay, so I'm taking this chance to explain to you because this is an important skill. All right, you need to know how to recognize which is unicot, which is monocot, which is udicot. Okay, first of all, look at the structure of stem, and you must also know how to label. So the one on the left is actually a monocot. Okay, and the one on the right is a udicot. Later, okay, not later, like after I tell you, yeah, how you want to recognize. Okay, it's very easy. First, label first. All right. Now we start with vascular bundle. You see all these entire oval shapes. All these are vascular bundle which contain xylem and phloem. So you have learned this actually in Form 3, right? The position of phloem and xylem. So if you were to look at a vascular bundle, the phloem is always nearer to the epidermis, okay, compared to the xylem. The xylem is always facing inwards. Xylem is facing inwards. Okay, so vascular bundle consists of phloem and xylem. All right, and then you will see this in between here, the red ring there, right? This is in between the xylem and phloem. There will be vascular cambium. Okay, vascular cambium. This is the lateral meristem. They will carry out mitosis to increase the number of cells, which is actually new xylem and new phloem. Okay, next. Now you'll find in the middle, this is called the pith. In DM, we call empulo. Okay, they are just parenchyma tissues. They store food. All right, the function of parenchyma tissue is to store food. All right, okay, and then cortex. These are the ground tissues that are, if they are not vascular bundle, they are not the epidermis, they're actually the cortex. In fact, the pith is also part of the cortex because cortex consists of, the parenchyma, collenchyma, or sclerenchyma. If the stem is young, if the plant is young, it's probably just parenchyma. When they're older, all right, they will form this. Some of them will have this sclerenchyma or collenchyma. Okay, and then cortex is over here, right, for the monocot. And now please don't forget to label the epidermis, and make sure when you draw epidermis, it has to be double layer. The epidermis is not just one line. You need to draw 2 lines to show that is the layer of epidermis. Okay, now how do you recognize monocot stem and dicot stem? How do you differentiate them? So you will see very clearly is the distribution of the vascular bundle. For the monocot stem, there's no pattern. The vascular bundle is scattered. Okay, there's no pattern. There is no particular arrangement for the vascular bundle in monocot stem. But for in udicot stem, you will see that they are very neatly arranged in a concentric circle, right? It's like a what you call, not concentric circle, sorry, in a circle, in a circular manner. It's all arranged very neatly in a circular manner, circular pattern. All right, so that tells you this is udicot. All right, okay, next, what about root? Okay, how do you recognize which is monocot and which is udicot? Okay, first of all, how do you know the root? Very easy. You can see sometimes when you draw, you see all this root hair cells. Root hair cells will be all this fine projection, all right, coming out from the roots. So now let's label first. Now, first of all, you look at the epidermis. Always remember you need to draw 2 lines to show the layer of epidermis. Then inwards, you will have the cortex. Okay, cortex. Then Further inwards, you will have what we call the endodermis. This forms like the what you call the steel. This is the inner part. Okay, so in the root there is like another layer, right? The core of the root there is this endodermis, and then you will see that inside the endodermis is another layer here called the pericycle. Okay, so here in this picture, endodermis is labeled a gray color, and the pericycle is labeled blue. Right, so that is pericycle, and now then you see the pink color one, the tissues that are colored pink here, they are the xylem. Okay, so you can see the arrangement of xylem in the monocot root and the dicot root, they are different. Okay, and the one that is green color, alright, those tissues are green color, they are phloem. Okay, later I'll tell you how to recognize them. Okay, next, root hair. Alright, so this is referring to the root hair that is jutting out here, and pith. Okay, so monocot root has pith. Right, the monocot root in the middle is like just the parenchyma tissues there. But when you see the eudicot root, no pith. Okay, to the central part, the middle part is the xylem. So you can see the xylem for eudicot is like an X shape, or we call T shape. All right, X shape or T shape or star shape. Sometimes you hear star shape, X shape, T shape. That is the eudicot root. The xylem is found in the center there in the shape of a T or an X, or we call star shape. Okay, and then for the monocot root, there's a pith right there in the middle. Okay, now what about the phloem? For the phloem for the eudicot root is found in between the spokes. You know, all this the T, you know, the spokes of this xylem right here. So let me draw here. This is your xylem. The spokes are here lah, in between all this, what you call this parts here. Alright. So not good enough. Let me put pink color. This is your xylem. Sorry, this is your phloem. This is your phloem. They are found in between the spokes, like the wheel of this of the bicycle, the wheel, right? Okay, and then the phloem over here is found in between the what they call the xylem over here, right? The middle part is the pith. So the arrangement of the xylem and phloem, they are different for monocot root and eudicot root. Okay, so monocot root has pith, but eudicot root has no pith. But when you talk about stem, remember the stem? Eudicot stem has pith, but monocot stem, no pith. Okay, so you can see the difference there. All right, okay. So remember, draw this. You know, you really want to know it, please draw it out in your notebook, label it. You may be asked by the question to draw. So you need to know how to label properly. Okay, all right. Now, so just to test you for a while, right? Is this a eudicot? Is it a shoot or a root? All right, or is it eudicot or monocot? Is it a monocot root or eudicot root or eudicot shoot or monocot shoot? Okay, so if you would like to answer, just type in the comment there. I don't know how many of you are watching. Okay, probably not many. All right, if you would like to answer, just try. Okay, just try. Okay, all right. I think I probably won't wait for you. Probably if you have you want to answer, you have answered already, or you have said hi, teacher. All right, so you do not want to be known. It's fine. So this is actually, you will see that the arrangement of the vascular bundles are very uniformly arranged in a circular manner, and there's a pith in the middle. So this is a eudicot stem. Okay, this is a eudicot stem. Now next picture. I have an answer here. I have a response here. Very good. Emlyn, all right. This is we're talking about stem, yeah. Okay. Oh yeah, stem now, right? It's a shoot, not shoot or root. Sorry, it should be. I'm asking about a shoot now. Not shoot. Sorry, stem now. It should be stem, yeah. But yeah, you already recognize it as a stem, yes. But further, you need to tell me whether it's eudicot or monocot, right? So how to see whether eudicot or monocot? You look at the arrangement of the vascular bundle. So it is neatly arranged in a circular manner like this. This is a eudicot stem. Okay, and Lynn, another picture. Right? Can you tell me whether this is a stem? Not sure, I've written wrongly. It should be stem. 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 Not sure, sorry. Let's let you Okay. And Lynn, you can try another one. Yeah. So what about this? So just give me two words. I want whether unichord or monocord, and then I want the word whether stem or root. Okay? Look at properly. Look properly. First of all, ask yourself, is there pith? Is there a pith? Because the existence of a pith will actually tell you whether it is unichord or monocord, whether it's a stem or a root. Okay, let me just check the answer. Root, all right. You said root is correct because you can see what? You can see the root hair cell. Yeah, that's easy. Now, how to see whether it's unichord or monocord? Okay, you've forgotten already because I already, I changed the slide already. So later you go and look back at the video and copy down in your note and make a mental, and remember it that which is unichord, which is monocord. Okay, tell me. Monocord. Yes, very good. This is a monocord root. Okay, so root because of the root hair cells, and monocord because it has teeth in the middle there. You see the empty space there, and not really empty lah. It's you have parenchyma cells lah, right? So here is the teeth, and you can see that alternate arrangement of the xylem, phloem, xylem, phloem, xylem, phloem around the teeth, right? The red ones or the pink one, that is the xylem. Xylem is always bigger in size, right, in diameter, compared to that group of what you call the seed tube in the phloem. So whenever you see, you're not sure which is xylem, which is phloem, always go for the bigger one, and it's more geometric shape, and it has got thicker cell wall. That is probably xylem. Phloem will have thinner cell wall. Okay, all right, fine, good. Arrangement. Yeah, the arrangement use alternate lah. Okay, and then alternate arrangement between the xylem and the phloem tissues around the pith. Okay, this is in the endodermis, in the middle, inside the middle part here. Okay, right, now we go to another part, which is primary growth and secondary growth. Okay, this is important. So every plant will undergo or carry out primary growth, which is growth of actually just, shooting up. The shoot will go up, right, to grow taller. And the secondary growth is not for all, right? So primary growth is for the shoot going upwards and the root going downwards. Okay, right, let's look at it. So here, if you were to label, right, remember all your apical meristem. This is the shoot apical meristem. This is the lateral meristem. This is the root apical meristem. Don't be mistaken. At the here also you have lateral meristem. It's just that it's not labeled lah. So here you also have lateral meristem in the root also, because there is the cambium. All right, there's also cambium there. Now, primary growth. Okay, what is the definition of primary growth? This is actually a process which involves an increase in stem and root length. That means, basically, it just means that your plant is growing taller. It is already carrying out primary growth, growing taller and growing longer into the ground, okay, into the soil. It occurs because of mitosis, that means mitotic cell division, which is happening at the apical meristem, that's at the tip of the shoot, at the tip of the root. That's how they can grow, because the number of cells increases, so it becomes longer up there and longer down there, right? So both ways, okay, vertically up and also vertically down, right? So it occurs in all plants. So all plants will be able to carry out primary growth. Otherwise, there's no use. The plant can't even get enough sunshine, and there's no root to go down into the soil to get water. Okay, so primary growth is carried out by all plants. Okay, now let's look at secondary growth. Secondary growth is not carried out by all plants. Okay, there are certain, usually monocots don't carry out secondary growth because they do not form woody stem. There is no bark, right? Normally monocots don't, right? Secondary growth is usually carried out by eudicots, and they form the woody material inside the inside the stem. All right. So secondary growth definition is a process which involves an increase, not increase in length, but sideways. That means the stem or the root become bigger in terms of diameter. It becomes fatter, like in other words, fatter lah, huh? Increase in the girth. Girth actually means diameter, right? Or circumference of the plant. This is also due to mitosis, but the mitosis is not taking place in the apical meristem. It takes place in the lateral meristem. So then you can see that the secondary growth involves the lateral meristem, that is in the cambium, right? The cambium tissues, the cord cambium and the vascular cambium in the lateral meristem, right? So you have it at the stem, you also have it at the roots. Okay, and it occurs mainly in woody plants. That means plants that are able to form wood. In fact, why it can form wood is because it already carry out secondary growth. Okay, so those plants that are herbaceous, that means they don't become wood. Like for example, the mustard lah, your sawi lah, okay, your kangkong lah, they don't form wood. Okay, they whatever you just don't eat it, it becomes old. And once it's old, it dies off. It doesn't become kayu. So it doesn't carry out secondary growth. Okay, right, next. Now cross section of eudicoट stem. Let's see how secondary growth happens. So first of all, you must look at the cambium. Where is the cambium? Secondary growth involves cambium. So you can see this orange line here, which is actually in between the xylem and phloem. That's the cambium. Okay, so after repeated what you call cell division, mitosis, you will find that that layer will expand. You will form new cells. You can see the red one. You get this pink color, which is lighter color. That is the new xylem. And on the other side, right, facing the outer side, you will find this light blue area, right, which is here, the light blue area. That is the new phloem. So it secondary growth actually forms new xylem and new phloem. And as a result, you see that it is being pushed upwards. The new phloem is pushed outwards, the old phloem is pushed outwards, the old xylem is pushed inwards. So it all causes an increase in the diameter. So the plant becomes fatter, in other words. Okay, so if you want to label, please label carefully. The secondary xylem is just next to the cambium, beside the old xylem. The old xylem is called primary xylem. The new xylem is called secondary xylem. Okay, and for the other side, you'll find the phloem. All right, the secondary phloem is pushed outwards towards the epidermis. Okay, for the secondary xylem is pushed inwards, that's towards the pith there. So you must know how to label properly, right? It's just beside the cambium, but it's just under the layer of the old phloem. All right, okay. Yes, any questions, just type. I will just go back, get back to you and try to answer questions. Okay, so you see, if you look at a picture to show that the secondary growth has taken place, you will see that the second picture has the bottom part here. You see this woody part, isn't it? The woody part shows that secondary growth has taken place. Therefore, it has become a bit, wider in terms of diameter increases. So how would you label? Okay, always remember there's epidermis outside there. Then you have the prime. This is not yet undergoing secondary growth. You can see that it's still very young. There's a young part there, right? Primary phloem is here. Vascular cambium is that in the middle there, the red color line there, and here's the primary xylem. Okay, so after secondary growth has taken place, you'll see that it becomes, pushed upwards. So the secondary phloem is the pink color one over here. It is just behind the primary phloem, which is the orange, the brown color one, right? And then the secondary xylem is the light blue here, and it's just next to this, what we call the primary xylem. The old one has been pushed inwards. Okay, and towards the epidermis side there, you will also have a layer of what we call cork cambium. It also carries out mitosis to produce cork. So these are new tissues being pushed outwards, and as a result, the woody stem, right, you will have a layer of cork under that the bark, right, in the bark. So here, give another year, right, probably older, right, you'll find that the layer will increase, the length, not length, the diameter of the stem increases. Okay, and it forms the periderm, which is a combination of the cork and the cork cambium, and also there's the bark there. So that's why you call the kulit kayu is actually bark, or it involves a few layers of tissues. Okay, all right. Next, now secondary growth. So after many years, it's a continuous, expansion. The diameter becomes increase, and you will find that in temperate countries, we have four seasons like spring, summer, autumn, and winter. They will form what you call annual rings. You can see dark lines and bright lines. Okay, these are called annual rings. Annual means it recurs every year. During summer and spring and summer, the growth rate increases. Growth rate is higher because there's sunshine, there's water, right? So therefore, you will find, all right, you have a lighter ring. So you see the light ring in your annual ring is actually formed during spring or summer, okay, where the growth is at a higher rate. And then the xylem that is used, the new xylem, that is actually like the cell wall is thinner, okay. And then dark ring is formed during autumn and winter. So you find that there's one dark ring and one light ring. If you combine it together, actually it shows 1 year of growth. One dark ring and one light ring is 1 year of growth. Okay, so you can count how many annual rings it has, and that shows you how many years the tree has been growing, or rather how old the tree is. Okay. Okay, so in Malaysia, it's not very obvious. The reason is because we don't have autumn and winter, like you said. We don't have autumn. We have only, it's like one whole year is basically a lot of water, a lot of sunshine. It rains like almost a whole year through, right? Unless you have specifically a very dry season, that is like, musim kemarau. Then probably for that year you will have a dark ring there. But usually in temperate countries, not temperate, sorry, in tropical countries like Malaysia, we don't have very pronounced the rings here, because we don't see the difference in the growth rate during autumn and winter. It's like the whole year is like wet and wet and wet, and sunshine, sunshine, sunshine. So it's like the plant grows even rate of what you call growth the whole year through. So the rings are not obvious if it's a tropical wood lah, right? Okay, understand? So if you only temperate countries where you have winter, you have very obvious difference, that you have a different rate of growth. Okay, right? Okay. So if any further question, you can ask me later. You can PM me and Lynn, yeah? All right, just want to finish up the slides here. Okay, importance of primary growth. Now, why do we need primary growth? Okay, this is very easy. So just I told you earlier that primary growth involves an increase in length in the shoot going upwards and the root going downwards. So it's very important. You need the shoot to be able to grow tall so that it can obtain enough sunlight to carry out photosynthesis to make food. Otherwise, the plant cannot survive. Okay, it's very important. That's why all plants carry out primary growth. This is very important for a plant. It will be able to make carry out photosynthesis. Okay, next, for the roots, it will enable the root to grow longer into the soil so that it can absorb more water, to search for water. Okay, especially if the plant is growing in a place that's not a lot of water. Maybe there's more water supply down in the deeper layers of the soil. That's why it has to grow downwards to search for water. Okay, next. And one more, right? It also provides support because by going downwards, it actually grips the soil. Okay, the root grips the soil. So it's important. Otherwise, the plant, when the wind blows, the plant can tumble, right? They can topple over. Okay, so that's the importance of primary growth. And secondary growth. Why do you need a plant to grow faster, to grow, in diameter, to become the stem to become, wider? Because if you only rely on length, right, the plant will be very tall and very skinny. Can you imagine a plant is very tall and skinny, right? There is no strength to support a very tall plant if it's very skinny. So when the wind blows, the whole tree will snap, right? And then that's the end of it, die already, right? So it increases the girth, or we call the circumference or the diameter of the plant. So as to provide greater mechanical support. You want to make sure the plant can stand upright. Okay, and for the stem itself, inside there, we just want to talk about the cambium producing new xylem and new phloem. That's called the secondary xylem and secondary phloem, so that you have more tissues to help to support to transport water and also food. Because as the plant grows bigger, the demand for transport of water and food will increase. So you got to make more tissues of xylem and phloem, okay, to meet the demand. Okay, next. And one more, produces cork and bark. You see at the, we call at the epidermis, just below the epidermis, you have this cork. All right. Now this is important as it serves as a layer of protection actually. Cork is waterproof. That's why I use cork for champagne bottles because it's waterproof enough. All right. And it is able to protect the plant from entry of pathogen or bacteria because the bark will actually crack. Sometimes because of the pressure, as it pushes outward, some of the bark actually crack. So when the bark cracks, it's dangerous because the bacteria can enter. When bacteria enter, it will destroy the tissues. It will make the plant have disease. So another layer, actually that cork here, is to protect the inner tissues, okay, from the infection of bacteria. All right. So now compare. The last part here, we talk about comparing between primary growth and secondary growth. So again, always when you see this question, don't forget to talk about similarities. Many students will go straight with the differences and they will lose marks. This is usually in essay questions. There will always be at least one question, one part of the question, either A or B or whatever, ask you to compare between A and B. Okay, so what are the similarities? So both types of growth occur in woody plants. All woody plants will have primary growth and secondary growth. Okay, but not all plants have secondary growth. But woody means it carries up both. Okay, and both causes an increase in the size of the plant permanently and irreversibly. That means it can't go backwards because it is a growth. You can't ungrow, isn't it? You have grown tall, you can't, shrink yourself back, isn't it? So this is actually a growth that you've learned in chapter 15 from 4. Growth is an irreversible change in the size, in the number of tissues, and so on, number of cells. Okay, and both involve mitosis. Yeah, okay, whether it's primary growth or secondary growth, both all this growth is due to mitosis being carried out by either apical meristem or lateral meristem. Okay, right, so these are the three major similarities. Okay, differences. Differences, I have 8 here, right? So remember, when you write your answer, please write primary growth is this, but secondary growth is that. Okay, write both sides and always put it in one sentence. Don't dedicate one whole paragraph on primary growth and then another paragraph on secondary growth. You will lose a lot of marks because this is not how they mark it in SPM. They need you to compare in one sentence. Okay, so differences. So involves apical meristem. What is the name of the tissue that is involved for primary growth, to carry out primary growth? Is apical meristem. Okay, and for secondary growth, it involves lateral meristem. Okay, this one you should remember lah, right? All this. Next, primary growth increases the length of plant, so it goes taller and longer, right? Whereas secondary growth increases the diameter or the circumference of the plant. Next, primary growth occurs longitudinally, that means vertically, upwards and downwards, right? Whereas secondary growth occurs radially, that means it goes sideways, it goes outwards. Okay, either the stem or the root, it goes sideways. Okay, next, forms cortex. Primary growth will make all these basic tissues, the cortex, right? Also makes the primary xylem and the primary phloem. But secondary growth will form all these secondary, these groups of tissues where you have the bark, you have the cork, you have secondary xylem and secondary phloem. Okay, modify. Primary growth does not form the tissue. So you will see that it's herbaceous. Definitely it doesn't undergo secondary. It's only undergoing primary growth. Whereas secondary growth will form living tissue. Okay, next, number 6. Primary growth Just a while, the slide is not coming yet. Okay. Occurs after germination. So after the germination is the process where the radicle grows out from the seed and the plumule goes out. That's called germination. That it will continue straight. Okay, it will carry out immediately after germination to make it taller. The stem will grow taller. Alright, but secondary growth only occurs after primary growth. So the basic thing of growing is actually primary growth. Then you have to grow first, you have to grow tall first, then only you go sideways. Okay, you can't be going sideways and then grow tall later. Right? It's only going tall, then you go sideways. Okay, next. Primary growth does not produce annual growth ring. We don't have that annual growth ring. Right? We don't have that annual ring. Right? Whereas secondary growth produces these annual growth rings. Okay? Primary growth is carried out by all plants, whereas secondary growth is only carried out by certain eudicots. That's why I say eudicot is certain, not all eudicots, certain eudicots. Most of them, yes, carry out secondary growth. Okay, so this is the differences. Okay, the last part here is the importance of the economic importance of plants that have undergone secondary growth. Now, this part, a lot of students will always answer wrongly. When you see the word economic importance of plants, that means it's not to the plant. The importance here is in terms of monetary gain. How is it important to human that we have wood? And all that. So it's not, when you answer economic importance of plants that have undergone secondary growth, is to provide support for the plant. Wrong. Because it's economic in terms of money, in terms of cash. Why is it important for us? Okay, for human. So be careful when you see the word economic importance is always for the human. What importance of this thing to the human? Maybe you can get more money, or you can sell it. That kind of thing is economic. So don't say economic importance of plants that undergone secondary growth is to provide support so the plant can support itself, mechanical support and all that. It's wrong. Okay, it's important to the plant, but it's not economic importance. It's only importance. Okay, now, so the economic importance number one, very important here, is basic. This secondary growth is the growth that produces wood. So it produces strong and hard wood. We call timber. Wood that we cut down is called timber. All right. So once we cut it down, this is called timber, and then we saw it, and then we can make important things like things that are useful to us: furniture, buildings, bridges. Example of important and very expensive wood actually is like kayu jati, called teak. Very strong, good quality. Mahogany, ebony. All these are names of certain species of wood, all right, which we get from trees. Okay, which have economic importance. They're very expensive actually. Okay, next. Besides that, certain trees or wood, right? Certain trees are woody trees. They produce resin. Resin is some kind of the sticky substance. When you hurt the tree, right? Some kind of liquid will ooze out, will flow out. That is actually resin. And sometimes are sticky. And sometimes can be poisonous, So this the orang asli, the indigenous people in the hutan, they will use it to hunt because this resin, okay, we call damak, which have poison, so they can use to hunt animals. So they shoot the sumpitan, sumpit, and then it's like a spear, right? Okay, resin and oil, they are commercialized, from the tree you can get oil, you can get perfume, you can get medicine. Okay, you get medicine. Yeah, some medicines I get from trees, right? Example, quinine. Quinine is to is a cure, it is a medicine for malaria and so on, right? For example, pine tree, merawan, these are Malay names, meranti. Kemenyan, kemenyan is, it smells good, right? People use it in to smoke, for religious purposes. Incense, we call it sandalwood. Tan hyong, I call it in Cantonese tan hyong. And also, like joss stick, when the people pray, play the Taoism, all this joss stick and all that, some have a very nice smell. They come from the trees. Okay, right. Next part. Now then, we're coming to the end now, don't worry. Okay, used as ornamental or decorative. Now, some plants are very beautiful. Like this is a plant that produces, a tree that produces very beautiful flowers. So we use it as ornamental. We use it as ornaments. We decorate our houses. And all these annual rings on the wood They are very attractive texture. So you see all these lines, very nice texture. Example of an ornamental plant is called bonsai. Bonsai is a Japanese art of planting it's a miniature tree. It's a small tree the what you call the type of tree is very small only. You can plant it on a small pot, but it looks like a like a woody tree. It's like very nice, beautiful. Okay, bonsai. All right. Okay, one more. Okay, and then these trees bear fruits. They have commercial value. Commercial means you can sell it, you can get money, generate income for the tree grower. Okay, though this tree, because they are woody, they can live many years. They live very long. They are able to bear fruits repeatedly, year after year. Durian, durian very expensive nowadays. You got a good durian tree, you can earn you a lot of income because they bear fruits every year. Right? Not only 1 year, you don't earn only 1 year, you can earn as long as the tree is there, it has got the nutrient, it will still bear you fruits. You can sell it, you can get money from there. Mango, rambutan, mangosteen, all this is economic importance to human because they have undergone secondary growth. That means they can live very long. They have woody stem. Okay, they have all these wood part. That means it can grow many years. They can live many years, and it's good for you. You can get repeated income every year. Okay, all right. Okay, so thank you very much for joining me for today's lesson. So this is hopefully is a revision for you for your exam next week, maybe your bio paper. So do questions, and if you are stuck, you would like me to help you to answer questions, no problem. All right, I give you my what you call Telegram name. All right, you are most what, most okay, so yeah, most welcome to message me if you want to ask me questions. Okay, so here's my Telegram name, and if you want to see more videos that I've already made, so go straight to my channel. Just type youtube.com/ my name is Pig Lee Chong, right? So you will able to see my playlist with many videos, hopefully, yeah. Okay, so you can't find anything you would like to find, you just message me and I can even send the link to you. All right, okay. So thank you very much, and I hope to see you another time. I do not know when I'll be making another class because all these slides need time to prepare, right? So I hope to make another topic soon, probably later. Yeah, okay. Thank you, Emily, for joining today's class. All right, so I'm going to end my lesson now. Please do get in touch with me if you need any help. Okay, all right. Bye bye, everyone.